Welcome everyone. So first off, before we have our main presenter, we'll have a short presentation from Julie Bonin on the Phragmites reporting tool. For some of you might remember her from last year on her presentation, so she's here to give us an update on that. And then for our main speaker, we have Dr. Amy Schrank. She's a fisheries and aquaculture extension educator with the University of Minnesota Sea Grant and an adjunct assistant professor in the Department of Fisheries, Wildlife, and Conservation Biology at the University of Minnesota. Her current research interests include understanding the effects of dam removal on stream fisheries and how invasive hybrid cattails affect fish communities in the Great Lakes region. Her outreach work involves collaborating with fisheries and aquaculture researchers and stakeholders around Minnesota to provide research support and a bridge to communicate technical information to stakeholders, managers, and the public. She has a BS in biology and Spanish and an MS in aquatic ecology and management from the University of Michigan. And she also has a PhD in zoology and physiology from the University of Wyoming.
Okay, perfect. Yeah, so I just wanted to give a quick update today on invasive Phragmites and how all of you can help us out reporting it. So we are still finding invasive Phragmites in the landscape. I have a new contact down in southwestern Minnesota, and this winter he found about 5 or 6 populations down there. So we do think that there is Phragmites in the landscape, invasive Phragmites that we're not yet aware of and we haven't yet documented. And our goal this year is to treat 100% of the populations. So the more we get reported, the more we can treat this coming season. So as you're all out in the field this year, if you can keep an eye out, that would be awesome. And I'm going to talk a little bit more about, how you can do that in a moment. So if you missed my presentation, which I think was the end of last year to the WPA group, just a quick introduction to invasive Phragmites. It's a very, very tall grass. There's a 6-foot tall guy standing there. And if you see something like this as you're driving around in the landscape or heading out into the wetlands, hopefully you'll take notice and be a little bit alarmed that this is not normal. We do have native Phragmites as well, and that's also very tall. But there's something about the invasive Phragmites that's even just a little bit more alarming than native Phragmites. So your first clue is that it's very tall grass. It has almost these bamboo-like stems and very large inflorescences. So the seed head or the flower head. Currently in Minnesota, our known distribution is just over 960 documented populations. And note that in 2019, we had 435. So the number has grown a lot. I would note that a lot of those new populations are along roadsides. Mowing seems to be spreading it pretty effectively, but a lot of them are out in wetlands and in other landscape situations as well. About 25% of populations are associated with wetland habitat, so that's a good percentage of them, and so that's why we definitely want to, inform persons like yourselves, professionals like yourselves, who can help us monitor areas that we're not going to be able to get to. In 2021 or previously, we got about 75% of all those populations treated. And so we're making good progress. But as I said, we hope to treat an even higher percentage of them this year, doing follow-up treatments on the ones that we've previously treated and doing initial treatments on those that we haven't gotten to yet. So how you can help is you can report invasive Phragmites for us. So you could download, if you haven't already done it, download the EdMaps Pro app on your phone or your tablet. And then if you find a population, you can just pull up your app really quick, and there's a little plus sign in the corner of the screen, and then you get to this reporting screen where you can add photos. The coordinates will pop in automatically, or you can create a polygon. You can add the images, select a habitat type that is appropriate, and then estimate the size of the population if you didn't create a polygon, so that we have a sense of how big the problem is at that particular location. You can also use the EdMaps Pro app. One way that is really effective is if you download the data set for invasive Phragmites, then when you're driving around out in the countryside or wherever you may be, if you happen to see a population of suspected invasive Phragmites, then you can pull up the app and you can see if it's already been reported, so that you won't double report it. So in this screen, it's not there, but your little car icon would be driving along here on Highway 55. And if I were next to this population, my little blue icon would be here. And I'd know that, yep, that one's already reported. If you don't see a big red icon like that, then it's something that we would want reported. And I would note that we are also documenting native Phragmites. There are some areas of the country where the native Phragmites populations, along with the rest of nice high quality wetland vegetation, has been just kind of decimated by invasive frag. So we are actually tracking native populations as well. In case we lose them someday, we'll have a record of, you know, what was the extent of native Phragmites in Minnesota. We hope that it won't come to that. But if you were to do that, you could just send me coordinates and some photos in an email, and I would track those on a separate map from EdMaps. So really quick, we do have native Phragmites in Minnesota, and we have the invasive Phragmites. And it's not actually that helpful to just look at these from the road or from a distance and be able to make a determination. There are many similarities, and they can be confusing, and we have had a few people mix them up. But once you learn a few cues, then you can distinguish them pretty readily. So on the left was the native Phragmites, and on the right, the invasive. So if you were to make a report, I would like to have 3 photos submitted with the report. And in particular, I would like an overview of the stand. In the picture on the right, this one is pretty clear to me from the inflorescences that it's invasive, but this one a little further off is not. I mean, they're kind of robust versus the sparse inflorescences of native Phragmites. But I'd want more information. In particular, if this picture were submitted, I'd not rely on that entirely to make a positive ID. So submit a photo of the stand, and then also a photo of the stems / sheaths. So the native Phragmites, recall, the leaf sheaths are loose, so we can see this gapping edge to the leaf sheath, they gap away from the stem. They don't overlap, so you can actually see little naked sections of the stem. And those little naked sections of the stem are red, glossy, and smooth. And these loose sheaths tend to fall off in the winter, so you still will have then a whole stem that's kind of naked, smooth, and glossy, and it will still have a little reddish blush in the winter as well. As opposed to the invasive Phragmites, which has these tight sheaths that overlap each other. So here's the top of a sheath below, and the bottom of the sheath just keeps going right down inside of that one. The sheaths are ridged and ridged like corduroy, so you can scratch those with your fingernail and feel that rough edge. And they tend to be overall green, and they remain intact in winter. So the stems are actually hidden beneath the sheaths, and you don't actually really ever see the stems. Sometimes this can vary a little bit in water. If invasive Phragmites is growing in standing water, then you may actually see a little bit of a reddish stem at the base of the plant. And here are a couple of photos that are similar to what I would love to have you submit if you're going to make a report. So on here, he was trying to show the ligule, which is another characteristic I like, but I can't really see the ligule there. But behind that, I can see these sheaths are intact in winter, and I can see that they're overlapping. So this is invasive Phragmites. And on this one, I can see that the sheaths have fallen off and exposing more of the stem with that reddish blush. And so this one is clearly a native Phragmites. So submitting a photo like that is really helpful. And then thirdly, the last picture to submit is one of the ligule. On native Phragmites, the ligule is this upright tissue. It's a little more than 1 millimeter tall, and it has a little bit of a ragged flap or edge to the flap of tissue. And there's a little fringe of hairs, very short hairs on the top of this little flap of tissue. There are some longer hairs as well, but they both have both the short hairs and the long hairs. The characteristic to pay attention to is the height of this little flap of tissue. The invasive Phragmites does not really have a flap of tissue at all that you can catch with your finger. It just has this discrete thin line, but still has that fringe of hairs on it. This discrete line is often kind of a soft V or a soft U shape, upside down U shape or V shape. And so where do you find a ligule? To find the ligule, you're going to grab a leaf blade and you're going to pull it back away from the stem. And right at the top of where the sheath joins the leaf blade, you'll find the ligule on the inside wrapped around where the stem comes out of the sheath. So on the inside, not on the back side. And when you pull that back, this is what you'll see then. And this is a really great way to take a photo like this, because a lot of times your camera won't focus on just one little piece of a stem or the sheath. But if you put 3 of them together, or put 3 of them on a clipboard or against your knee or something like that, some surface, your camera will focus better. So this is a nice image of ligules of an invasive Phragmites, and is really helpful for me making a determination remotely from a photograph. Okay, so that's basically what I had today, and I'd be happy to take any questions if any of that wasn't clear. And definitely please feel free to reach out to me individually at any time.
Thank you so much. No questions are in right now, but for everyone who wants to have reference, you can see Julia's full presentation on the Minnesota Wetlands Professionals Association's YouTube page. This one will also be up for the recording. And I think there's one question that just came in. How do we download the data set in the app?
Oh, you have to go into EdMaps, and it's a little bit of a process, but they have a tools and training page. So there's a link at the top of the website online, and you can do the tools and training, and it will tell you how to walk through downloading those data sets. And if you have any questions, again, about that specifically, because you're trying to do it, reach out to me and I'll help you with that.
Great, thank you. It's probably a good group of people to present this to, so hopefully they can get that reporting out there. So anyways, well, thank you so much, Julia.
Thank you ahead of time for your work.
Yeah, great. So thanks again. And I think we will move on along to from Phragmites to cattails. Phragmites to typha. Here you go. So Amy, go ahead.
Great. Thanks, Jack, and thanks, Casey, for the introduction. So my name again is Amy Schrank. I'm a Fisheries and Aquaculture Extension Educator with the University of Minnesota Sea Grant, and I am going to talk to you today about cattail removal for ecosystem enhancement. And in my talk, I'm going to talk to you about a bunch of different projects, both in the Great Lakes, and then I'll talk in detail about our project that we have going on right now in Minnesota. My major collaborators on these chunk of projects are Shane Lishua, who's at Loyola University in Chicago, and then Dan Larkin at the University of Minnesota. So before I jump into the research, I just want to let you know that this is the small section of a larger couple of projects. So as you saw on the previous slide, they're funded by EPA GLRI money and also MACERC and Environment and Natural Resources Trust Fund money. So lots of folks have worked on this. The GLRI project was part of a bigger project that looked at cattail effects on things like birds. We did some work on amphibians and macroinvertebrates. I'm going to kind of show you a little section of that on fishes. So these are the collaborators and some of the organizations that were involved in some of these larger projects. It's really fun to work on, actually. So I'm going to start here with a review, which all of you will know well, of what a healthy nearshore lake ecosystem looks like. So in this photo, you can see a variety of shoreline plants. You see the occasional cattail here. When you move into the water, you see these sort of brown-topped emergent plants dominate. Those are, of course, bulrush, which is a really important species in the emergent zone in the Great Lakes region and in Minnesota as well. Of course, you move out into open water, you get floating leaf plants, and you get these submerged plants. So this is kind of what a healthy nearshore lake zone looks like, and you can keep this in mind as we move a little bit forward here. When we teach about this, I teach a lot of aquatics courses, we tend to teach that this is like a linear pattern, right? First you have emergent plants, then you have floating leaf plants, then you have submerged plants in the littoral zone. But from a organism's perspective, this really is different. Emergent plants are always mixed in, often mixed in with submergent plants and floating leaf plants. So you get this real complex of combined physical plant types. And it's this complexity and heterogeneity in plant growth forms in nearshore lake regions that's particularly important for fishes. They depend on these structures for protection. From a fish's perspective, things like bulrush are great because they don't accumulate a lot of litter year to year. They provide protection for fish from other fish predators, but they're thin enough that fish can swim through relatively easily. Then you have the presence of floating leaved and submerged plants. The leaves help protect fish from a variety of avian predators and also fish predators. And also these diverse plant structures provide habitat for fish food, right? Macroinvertebrates, bugs, other things that fish like to eat in these near shore zones. So that vegetation is important to hosting basically their fish diet. I have a grad student who thinks of it as the fish pantry. So it's really the variety and diversity of native plants that's important here in the near shore zone, especially when we're talking about fishes and macroinvertebrates. As you also probably know, from a fish's perspective, these nearshore or coastal wetland zones are important spawning and nursery habitats where small fishes are protected from predators, but they can find a ton of food to eat. And you can see here these photos. This is a spawning brook stickleback. We've got a couple of larval fish here, which spend a lot of time in these nearshore zones. And this is a baby northern pike, so they spend time in these wetlands as well. Most fish use these wetlands at some point in their lives. There's also species that live in these nearshore areas for their whole lives, so things like minnows. They spend all of their lives in these nearshore zones. So you have here red-bellied dace, blunt-nosed minnow, golden shiner, and common shiner. We don't often think of these if we're not really fish people. These tend to be really important as forage fish for the sport fishes that people tend to care a little bit more about. So pike, smallmouth bass, of course, we're in Minnesota, so walleye is super important, and yellow perch. They depend on these forage fishes for their food. So that's another reason this nearshore zone is super important. So what's the deal with cattail? As I'm sure you know, over the last few decades, lakes and wetlands in Minnesota and in the Midwest overall have seen really increases in invasive cattail. So the wetlands and nearshore zones of many lakes can start to look like this photo, which is, as you can see, is a homogeneous stand of invasive cattail. And it's really this homogenization that's a problem for many species. I showed those photos in the beginning of lots of different types of plants with lots of different types of growth forms. And so this homogenization and sometimes terrestrialization of aquatic habitat is really a problem caused by typha. So what I want to do is back up and talk about invasive cattail and where it came from. Again, this might be information you already know. But in Minnesota, we have, of course, the broadleaf cattail, which is our native species, Typha latifolia. We also have the narrowleaf cattail, Typha angustifolia, which likely came from Europe sometime in the late 1800s. And I'll talk about how that invasion happened in a minute. One quick thing about telling these species apart. So you can obviously, by the name broadleaf cattail, narrowleaf cattail, they have different width of leaves. So it's very hard to tell unless you really spend a lot of time looking at cattail and if you have both of them close together, so you can compare the widths. But you can also look at the sort of male and female parts of the plant. In the native species, these are touching. So that yellow top and the brown fluffy bottom are touching each other. In the narrowleaf cattail angustifolia, you can see here there's a space, right, between those two parts of the plant. This gets a little bit complicated when we talk about hybrids in just a second. So what happens in areas where broadleaf cattail and narrowleaf cattail coexist, they almost always hybridize. So what you end up with is hybrid cattail or Typha X glauca. In terms of plant identification, it can be a little bit more difficult. You can see there's a variability in that space between the male and female part of the plant. And there's also a lot of back crossing that occurs. So sometimes in clones where you have, actually it's a hybrid cattail clone, you'll have some stems where that male and female part of the plant are touching. So one of the things that you also kind of need to notice if you're trying to decide if you have hybrid cattail or invasive cattail or the native broadleaf cattail is also how it's growing. Our native cattail does not tend to take over. It plays nicely with other plants. It doesn't grow in a huge monoculture stand. So if you see only stems where the male and female parts of the plant are touching and it's not growing in a huge monoculture, they're not other stems where there's that separation, you probably have native cattail. If you have a huge clone and you have some stems with that space between those two parts of the plant, and you might have the occasional stem where those two parts touch, if you have that variation, you likely have a hybrid. So people have done work where field identification can be notoriously difficult. So it's something to be careful about. There's also genetic methods to tell, but you can sort of get an idea in the field as well. So where did it come from and how long has it been here? So there are extensive herbarium and pollen records that suggest that the narrowleaf cattail angustifolia showed up on the East Coast here around the mid to late 1800s. We really didn't see it inland at that time, so inland records really don't show that European species, but slowly it sort of marched across the U.S. By 1949. That Angustifolia had moved across to the Midwest. The red star is Minnesota, so it had gotten here already. And then you can see in '67 a little bit more expansion, and currently as of 1999, right? This came from a paper by Sue Gladowicz et al. In 1999. It was already expanding up into Canada, and it has continued to spread. So it's kind of a sneaky invasive species because it's been here a long time. It looks quite similar to our native typha. And there's not a great chance that we're going to ever eradicate it, right? It's pretty ubiquitous. So when we talk about what we're going to do about it, it tends to be control, not eradication. So that's important to keep in mind as well. So why is this hybrid such an issue? I just thought I'd give you a little background about that. In terms of species characteristics for all typha species, they have wind dispersed seeds. So you can see in this picture, you see these little tufts here. They also propagate clonally. You're all wetlands people, so you know the rhizome underground. They can also reproduce that way. So that's true of all typha species. The reason given for why this hybrid tends to be such a problem is something called hybrid vigor or heterosis. It's the idea that the offspring of two parent species does better than the parent of either species. So a classic example is in corn. You have parent 1 and parent 2, and the ear of corn from the hybrid is just bigger and more robust. And there's some evidence for this in typha X glauca, or the hybrid cattail. So it does have a more rapid growth rate than either of its parent species. It has a larger stature, just grows taller. This is my favorite example of this. So these are 3 average-sized human women standing up in a coastal wetland in northern Lake Huron. You can see there's some sedges and probably the occasional bulrush in the foreground here. The darkest color, right, is obviously forest, but this medium green is a stand of hybrid cattail that's been marching across this coastal wetland over the last decade or so. And you can see how much taller it is than all the rest of the vegetation around it, and also taller than these women in the foreground. Because it's so big, oh no, sorry, it forms larger clones as well, so it tends to spread out and form large clones. You can see a clone here in this picture growing and spreading. Because of its height and the larger clones it produces, it accumulates much more biomass. And this is one of the really big problems with this hybrid cattail. To describe this, if you've never walked around in a cattail stand in a lake, these are some researchers in this photo with 2 people. They're in a sort of native nearshore zone. You can see there's some bulrush, there's lily pads. It's easy to see. You can float your equipment. It's like a nice place to work, sunny. But when you get into a cattail stand, you can't see anything. It's very easy to get lost. It's definitely taller than your tallest technician, and you really have to shove things through it. It can pop your floating boats, and it pokes you in the eye. It's a very different habitat than these native coastal wetlands. So if it's a different habitat for humans, it's likely also quite a different habitat for fishes. The other thing, it's quite difficult to walk around because there's so much dead litter as well. So more biomass. It's tolerant of a wide range of water levels. So as we have as humans, we've made things pretty convenient for invasive species, and that's true with hybrid cattail as well. It tends to have a wider range of tolerance than either of its parent species, so it can grow in shallower water and deeper water than either parent species. It does really well in managed systems. So if you want to get rid of hybrid cattail, the flood, drought model is great, but we tend to not like that. So for example, in prairie pothole regions, hybrid cattail take hold when you have managed water levels where you don't have that flood drought system in place anymore. And that's the same in lakes in Minnesota as well. It also prefers nutrient-rich conditions, so it does really well, better than other plants at sequestering nutrients. And lastly, it's fairly tolerant of saline conditions. So we talk a lot about road salt and how that affects communities. It turns out that actually the Angustifolia, the narrow-leaf cattail that came from Europe, is the most tolerant of saline conditions. It can actually grow in sort of brackish marshes. Our broadleaf cattail is the least tolerant. The hybrid is somewhere in between. So it does tend to do pretty well in saline conditions. So that's the background of TIFA. And what I thought I would do is sort of tell the story of what we've been working on with hybrid cattail, both in the Great Lakes region and now in more specifically in Minnesota. And so what I've been focused on is how cattail affects different biota. So there's been quite a bit of work in the Great Lakes. And Shane Lishua and Nancy Tuckman's group at Loyola University Chicago has done a lot of this work, along with Dan Larkin at the University of Minnesota, about how TIFA outcompete. And when I'm saying TIFA now, I'm talking about that hybrid, right? The invasive hybrid. I definitely will forget to say that every time. So just know that that's what I'm referring to. But there's been quite a bit of work on how it outcompetes other plant species. It sequesters nutrients. It can affect the management of it can actually affect greenhouse gases. But there are fewer data about how TIFA affects other biota, including fishes. So there's a little bit of work on macroinvertebrates, but this big project we were involved with looked specifically on how management of TIFA affects fishes. So before you can measure how management affects fishes, TIFA management, you need to know how TIFA itself affects fishes in the absence of management. So that's sort of where we started. Our initial research compared native coastal wetlands, so you can see here in the photo labeled native, to hybrid cattail invaded coastal wetlands zones. So we were looking at different zones within the same wetland. And we were really interested in how water quality and fish community differed in these regions. And I'm not going to talk a lot about the methods for the Great Lakes work, so if you're interested, just feel free to ask me, but just in the interest of time, I thought I'd just talk to you about sort of the most interesting results we found, and then I'll dig a little bit into the details more for our Minnesota study. So for this sort of comparison of native versus invaded sites, we looked at plants, of course, and just to give you a sense of that, I'm going to show you data for Carex species, or how many sedges there were in each of these native and invaded zones, submerged plants, TIFA, of course, hybrid TIFA, and then just overall leaf litter. So when we looked at just percent cover, so abundance of these different types of plants, native zones here are in the blue bars, cattail invaded zones are in the orange bars. You can see, and it's not super surprising, right? We see lots more Carex species in the native zones. We see a trend of increasing submerged plants in native zones, but this difference was not statistically significant in our study. Of course, there's more TIFA. I mean, we were choosing TIFA zones, right? And also, importantly, a lot more litter, right? We know that TIFA produces a lot of litter, and this can be the source of some of the problems for hybrid TIFA. So there's significantly more litter in TIFA zones compared to native zones. So that's just kind of a brief summary of how the plants differed. We also looked at sort of water quality or environmental conditions. And so here I'm showing you water temperature, which is on the y-axis, and we have time of day on the x, so 10 a.m., noon, 2 p.m., 4 p.m. And this is just a plot of sort of all the temperatures throughout the day. We know temperature changes throughout the day as the sun warms the water. The black dots here are cattail invaded regions, and the open dots are native coastal marsh regions. And what you can see is, of course, in both zones, temperature increases throughout the day. And these pictures are just to remind you what these two zones look like. But you can see that temperature is consistently a couple degrees warmer in these native zones compared to the cattail invaded zones. And that's just an artifact of the shading, right? These cattail are so tall, they shade the water from the sun, so the temperature is a little bit lower. This probably doesn't have it probably has a variety of different indirect effects. It can affect things like fish growth or organism growth. It probably does affect photosynthesis, although no one's really looked at that, maybe algae. So that would be something interesting to look at. But there is a difference in temperature in both of these zones. More sort of immediately important for things like fish and even macroinvertebrates is dissolved oxygen in the water. So I'm just showing you some results with dissolved oxygen on the y-axis, and then cattail invaded versus native zones. What you see here is, on average, dissolved oxygen was significantly higher in native zones compared to cattail invaded zones. So this could be a mechanism for why some of these fish might be affected. If we dig into this a little more, you as wetland people know that probably that dissolved oxygen in the water tends to be lowest in the early morning after, photosynthesis has not been occurring all night. So there's definitely a low, and we see that when we look at these data across the day. So again, dissolved oxygen and time of day again here on the x-axis. So it starts low, it tends to increase more quickly in native vegetated regions compared to cattail invaded vegetated regions. So we're seeing more dissolved oxygen in these native zones compared to these invaded zones. You can see we're missing a little data in the afternoon, and we're still looking at this in some of our sites in Minnesota. So this dissolved oxygen seems to be a big issue. It would be very important for things like fish. So you have a lot of variety in oxygen tolerance for low oxygen in fishes. So this top picture is a mud minnow. These guys can actually gulp water from the surface when there's really low oxygen, so they're tolerant of low oxygen. But things like yellow perch are not tolerant, and if oxygen is low, they'll leave the area. What we think is happening with cattail, and we're going to look at this a little bit in our Minnesota study as well, is that, as we said, both sites have a low in the morning, but what happens in the normal course of a lake is you have wave action or currents bringing some of this still oxygen rich water from the pelagic or open water zone into these interior marshes. And if you have sparse vegetation like you would if you had bulrush and submerged plants, it probably gets this oxygen rich water into these interior marshes more quickly. Whereas when you have that dense live standing vegetation and dead litter in a cattail region, that oxygen rich water is just not getting through. Those waves are not propagating through that dense vegetation. So it takes longer for that oxygen to rebound. So cattail seems to decrease pelagic littoral mixing. So pelagic being open water, littoral just meaning the near shore zone. Those areas are linked, and cattail seems to really disrupt that linkage, which I think is important. Thinking about the effects for the ecosystem. Lastly, for this Great Lakes, our first part of this Great Lakes data, we looked at fish community and we looked at a lot of different metrics, but I'm just going to show you abundance. And again, the native zones in the blue and the cattail invaded zones in the orange. You can see significantly more fish, higher species richness as well in the native compared to the cattail zone. So this was sort of our first cut. So we can see that, yes, cattail does seem to have negative effects. It changes environmental conditions, reduces oxygen, and it's associated with reduction in fish abundance and diversity. So what are our options here, right? What can we do? Knowing that cattail is pretty ubiquitous, we're not going to get rid of it all. How can we handle it? So there's a lot of ways, as I'm sure you know, that we manage cattail. We herbicide it, we sometimes burn it, we harvest it above water, and sometimes we harvest it below water. Herbicide and fire can have detrimental effects on things, especially on things like amphibians and larval fishes. And so given how important nearshore zones are for these species, it's probably not the best management strategy. There has been some research into mechanical harvest. So for example, in the Great Lakes, region, there's data that suggests that this smaller local control scale of management can suppress typha. So things like cutting a channel through the dense typha can have human benefits as well as ecological benefits. And so that's one of the things we're looking at. So as I'm sure you know, well, first let me show you the comparison here. So this is what we were working on in the Great Lakes, and I'll show you some data here in a second. We were cutting these channels through cattail as one of our management treatments. And what you find out is that that native seed bank is still in the sediment. So once you get rid of all that, the cattail litter and the live cattail, you give space for these seeds to get sun and then actually grow relatively quickly. So we saw this a lot in our channel plots where you get native vegetation growing back. So what happens is, of course, as you know, the parenchyma in the stem of a cattail allows the oxygen to move to the roots efficiently. When you cut the stem below the water, it basically drowns the plant. Now that of course doesn't kill the clone, but it does prevent those stems from shooting back up again. So it does provide a few years of open habitat. And let me just go back here a second. So those the cattail, of course, eventually, you can see they eventually grow in from the side. This is, I think, 2 years after our initial cut. So you would have to sort of provide some maintenance if you're going to keep this open, but they don't regrow from the center. So this seems to work pretty well. So our overall question then is, how does mechanical harvesting of cattail stands affect coastal wetland systems? So I'm going to show you just a little bit of data from the Great Lakes and then talk about what we're doing in Minnesota here. So in terms of the Great Lakes work, we had we kind of used a block design. We studied this in 4 wetlands, and you can see there we're right again in sort of the tip of the lower peninsula of Michigan, in really mostly northern Lake Huron. Each of our wetlands was a block, and then we applied these 3 treatments in each wetland. So we had a harvest treatment control, which was nothing, right? You just cattail, and then this aquatic channel treatment. This harvest was cutting cattail in the late summer, so there's still standing water here. This is a special like amphibious harvester from the UK, but it was cutting cattail above the water, and then the channel was cutting the cattail below the water. So just to show you what we found with these treatment results, and I'm going to show you some data. We analyzed these data with linear mixed effects models. I'm happy to talk to you about the data analysis if you're interested, but I'm just going to kind of show you some graphs to give you the overview of what we found. So this first one's just a quick summary of maybe the most interesting results with plants. So here you look at the percent cover on the y-axis, and remember we had control, harvest, and channel treatments. The blue bars are submerged plants, and the letter, if the letter is the same, there's no statistical difference between those bars. So you can see that for the control and harvest, there was no difference in submerged plants, but we of course saw a difference in the channel plot. So quite a bit more percent cover of submerged plants in the channels compared to the control. So this and harvest. So this is just an artifact of opening up the space and letting those native seeds regrow. The other thing we looked at, among other things, was below water leaf litter. This was significantly different in all treatments. So it's the highest in the control, it was lower in the harvest, and the lowest in the channel. And so this has some important ramifications for fishes. So the only other thing I'll show you about is 2 other graphs from these data. Fish abundance, again, these pictures just reminding you what we did here. We saw differences in fish abundance in all 3 treatment types. So we saw significantly more fish in both the harvest and the channel relative to the control, and more fish in the harvest relative to the channel. So our treatments did have some effect on fish community. We also looked at diversity and richness and saw some differences there as well. This is the only time I'll talk about macroinvertebrates, but we did look at macroinvertebrates with this as well. And we did see an increase in harvest plots relative to the control. The channel, we had a tendency to have higher abundance of macroinvertebrates, but this was not statistically different than the control. But we were seeing some differences in macroinvertebrates. We looked at tadpoles too, actually, and saw a trend for increasing tadpoles in the channel, but again, nothing statistically significant. So we really are seeing a positive effects of our treatments on fish and macroinvertebrate community. So we're seeing this, maybe this can have a benefit. One thing about this harvest treatment, thinking of transitioning this work to inland lakes in Minnesota, it was hard to do this, right? Like I said, it was a special sort of amphibious harvester. It was always in the shop getting fixed. It was difficult to do this. So when I talk to you, when we look at the next chunk of our data in Minnesota, or next chunk of our project in Minnesota, we were really looking mostly at the control, only at the control and channel treatments, not the harvest treatments. Although I will say that one thing we sort of tried this past winter for a variety of reasons was actually cutting cattail after the ice forms. So that's almost similar to a harvest treatment. It would be interesting to look at that in the future. We haven't really looked at that in great detail, but that would, I think, be an interesting thing to look at, especially in Minnesota. So why do we even want to look at these channels? If we zoom out, channels are particularly relevant to inland lakes in Minnesota. So if you take a landscape view across Minnesota, we are making these channels all the time. Landowners use channels to gain access through cattails for their boats, for their view, for swimming, for lakeshore access. They typically dislike cattails because they're so dense, so it's hard to see. It's hard to get through cattails to get to the open water. Right now, as a landowner, you can get a permit to remove a swath 15 feet wide from shoreline to open water to remove cattails and whatever other vegetation is there, so that you can access open water from the shore. So in addition to that, evidence from our Great Lakes coastal wetland work and sort of thinking about these channels as something that already exists, we are wondering, are these channels benefiting? Are they providing some benefit to fish and plant communities and perhaps macroinvertebrates? And is it that, and can we use these channels to sort of try and regrow these native plant communities and restore some of that diversity in physical habitat in these lake zones? So that's what we're focusing on here in Minnesota. So I'm going to talk about our specific questions for our study, and I'll talk a little bit more about what we're actually doing around the state right now, because we're in the middle of it. We're in the middle of our project right now. I won't be able to show you final results because we won't get those until after this summer, but I'll show you what we've been doing. So our specific questions, similar to what I said before, does mechanical cattail removal increase plant diversity and benefit water quality in fish communities in nearshore lake ecosystems? So this is a picture that came from an aquatic plant management person from the DNR who said, anecdotally we see this, right? You see a boat, this is a short little channel. You can see this hybrid cattail stand here. And when you excavate these channels, even though presumably this boat is going in and out, you see some regrowth of native plants. So the idea is, can we see that in some of these channels? Can it benefit ecosystems? And the second question is, how does this vary across regions in Minnesota, right? So you can imagine in the north where lakes are pretty intact, the seed banks may be super intact. And if you remove cattail, maybe you will get native vegetation regrowth. And you need vegetation there for a variety of reasons. An important human reason, right, is shoreline erosion. You don't want to exacerbate erosion by removing all these plants if those are the only plants that are there. So we're hoping to get a regional picture of this as well by sampling lakes across Minnesota. So here are our study lakes. We've chosen 9, a couple way up north, Rainy and Cabotogama, some in the middle, and then a couple of metro lakes. We don't have a lot of southern lakes. We searched but had a hard time finding lakes in the south, so I would say that's one gap in our data at this point, but we do have 9 lakes to start. And to just describe our experimental design a little bit, so at each of these lakes, we've selected 2 paired sites. One site, and we've selected these sites to be as similar as possible, so they're in cattail invaded areas. And one site is where we will, cattail will be retained throughout the study, and one site we will remove cattail. These sites are 10 meters wide or 30 feet. We chose this width because it doubles the current landowner permit allotment. So you can have a 15-foot wide swath, and so this is twice that, 30 feet wide. We're going to sample, we have sampled both of these sites before cattail were removed. So this is a Backy design, right? Before, after control impact. Our impact is cattail removal. So we sample both these sites before we remove cattail. This happened last summer. We removed cattail at one of these 2 paired sites this fall. We are headed back out this upcoming summer to resample these areas using the same methods. So this is just a aerial photo of one of our sites in Big Marine. So here's our cattail stand. So we have 2 plots in here, and we'll remove cattail at one of these plots and come back and resample them this upcoming summer. So just a timeline, just to be clear, summer 2021, we measured water quality, plant community, and fish pre-cattail removal at both sites. In fall, we contracted out to remove cattails, again, below the water with mechanical harvest. And then we will be back at these sites in the summer to measure the same variables again. Just to give you a little bit of detail about the methods that we're using, this big green rectangle is just sort of a zoom in of one of these sites, both cattail retained and cattail removed. We divided our each site into 10 subplots. That's what these little squares are. And you can see up here is shoreline. I'm sorry, down here is shoreline, and up here is open water. And at each of these subplots, we'll be measuring ecosystem variables. So things like dissolved oxygen, temperature, conductivity, and pH, basic water quality variables. We'll also be measuring the plant community. So we'll be looking at species richness, abundance, percent cover, etc. Within each of these subplots. We'll also be taking a look at the broader plant community in the lake, but for the purposes of this study, I'll just talk about the plant sampling in each of these sites. We'll also sample the fish community. You can see those little rectangles change to these hash circles, which is to remind me to tell you that we use minnow traps to do this. It can be really complicated to sample fish within a cattail stand. So a lot of people set nets just outside of a cattail stand and assume that those fish that they're catching are the fish that were in the cattail stand, but fish are really good at using edges. So we were very interested in actually sampling within cattail stands. And so we used minnow traps. So as you know, then, as you can see by the holes in the minnow trap, the fish we're catching are usually, they can be adult minnows, but they're juveniles of other species. So we're, again, looking at things like fish community, abundance, species richness, diversity, and community. So those are our methods. We did successfully, despite it being kind of a weird summer and COVID and a strange year, we were able to do our sampling and also remove cattail at our 9 lakes. Actually, in Rainy and Cabatogama, we have 2 sets of sites. So we have 11 sets of paired sites total. And so we removed cattail at one of these 2 sites. And I show you these pictures because it's really different habitat, right? When you remove that cattail compared to the habitat around it. Just a word about our collaborators for this Minnesota project. We've had some strong support from the Minnesota DNR, Shane McBride, who's the Aquatic Plant Management Coordinator, and also Donna Dustin, who's retired, but she's a fisheries research biologist out of Detroit Lakes. We coordinated and sort of put together a lot of meetings with DNR about this, because one of the concerns with hybrid cattail and saying, hey, let's remove cattail is that, okay, that's what everybody's going to want to do, right? So we really have a focus on restoration of native species rather than just removal of cattail for removal of cattail's sake. So we've had some good collaborations and discussion with the DNR, really statewide. They've helped us choose sites because we're doing sites mostly on public land, and that's been a really good collaboration for us. We're also collaborating with Voyageurs National Park. Reed Plum, who's a wildlife biologist there, he has also actually since left the park for, I think, Superior National Forest. But he was helpful in sort of finding suitable sites for us in both Rainy and Lake Cabatogama. And they also did the cattail removal for us. So they've been doing a ton of work on cattail and Voyageurs. So he just sort of added our sites to his cattail removal plans, and they did those removals for us, which was really helpful. So that's been great. The graduate students on this project, Brendan Nee here writing on the data sheet. They're both graduate students in the conservation sciences program at University of Minnesota. And Brendan is heading up the fish component of the project, and Mike Tuma is heading up the plant component of the project. So they're out there in the field together, working hard. You can see it's pleasant when it's sparser. It can be difficult when it's dense. And then we had Kaylin Schmidt, who she's an undergrad at UMD, working with us on this project last summer. So they're doing the lion's share of the work in the difficult cattail marshes. So I thought I'd show you just a little data. I mean, we don't have a ton of data because the more interesting data comes after we sample once the cattail have been removed, but at least I can give you an idea of what we've found so far. We collected about 7,000 fish from June 30th to August 21st of 2021. And I'm just showing you simply total fish abundance here, so nothing fancy. These are our 9 lakes. And I'm showing you the bars for the control site and the removal site, but remember the removal hasn't happened yet. So what we kind of hope for looking at these data is just that their order of magnitude of number of fish is similar, and it looks pretty good, right? These are our northern lakes, Rainy and Cabatogama, our sort of middle and middle western lakes. You can see First Crow had really low fish abundance, and then our metro lakes. So not really a lot of conclusions here, just we caught a lot of fish. And you can see our fish are mostly small. I thought it might be interesting to talk about what kinds of fish live in these really dense cattail stands. So I've combined, this is across all the lakes, right? All the fish we collected, and you can see they're overwhelmingly sunfishes. So these are things like green sunfish, bluegill, pumpkin seed, smallmouth bass, largemouth bass, occasional rock bass. I think I have a picture. Yeah. So these are by and large what's most commonly in these cattail stands. And this is what we saw in the Great Lakes as well. These are fish whose strategy to escape predation is to hide in vegetation. So they will hide here. Now in the Great Lakes, we did see these in cattail stands, but we saw more in native stands. So I don't think they're choosing cattail stands over native stands, but they're using them in any case, whether or not they're ideal. We also see a lot of bullheads. So these are black bullheads. The one thing about collecting bullheads in a minnow trap that's important to know as a species, they have an interesting life history. The males guard the babies, like the small bullhead, for a certain amount of time. So they all swim around in this dense ball. And so if you're going to catch a few bullhead babies, you're going to catch a lot of bullhead babies. So we tend to see large amounts in our minnow traps. So we try to take that into consideration. We do see some yellow perch. This is driven largely by Lake Cabotogama. And we also saw mud minnows. So these are not actually a true minnow, but they're that species I talked about earlier that's really tolerant of low oxygen conditions. So you can see in this graph, I'm up to 6,000 here, right? This is a lot of sunfish. We did catch some of these other species, but I'm just shifting the scale on the y-axis here to show you what else we caught. So you can see a lot of sunfish, a lot of bullheads, a lot of mud minnows, a lot of yellow perch, which I already talked about. We also see banded killifish. So these look like minnows, but they're a surface feeder. We see generally some minnows, but not very many, and I'll talk to you about that a little bit more in a minute. We see some bowfin. I know these as dogfish. I don't know actually if there's another name for them, but we see these a lot in vegetation. Northern pike, of course, we see babies occasionally in these cattail stands and in native marsh as well. So that's what we're collecting. What I wanted to talk about briefly at the end here, I'm almost to the end and would be eager to take questions. But I want to show you some of the variation we see within cattail stands, because I'm particularly excited about this when we come back to sample this coming summer. So I'm showing you an example from Big Marine Lake. And this is just a diagram that Kristen Lobeek of Macer created, just showing you kind of what this is going to look like. We'll have a control site here with no removal. This would be our removal site. Probably these are shifted over, but you get the idea. If we kind of look at an overhead view of this, we have these two sites, which in reality will probably be more separated than this, but and you have shoreline and open water. And so I just want to show you some dissolved oxygen and fish community things that we're seeing so far and what I'm eager to find out about next summer. So you get the idea of what a site looks like. We are measuring dissolved oxygen, so this is average DO on the y-axis, along this transect. So we measure it within the cattail regions, kind of close to the open water. We measure it mid-stand, and we measure it near the shoreline. So this is all done underwater, right? So if we look at those data, and this is averaged across all of our sites, for a control and say our pre-removal plot, right? So this is our cattail remove site, but the cattail haven't been removed yet. What you can see is that DO does vary across, so from shoreline to open water. So you see the open water here is gray, mid-stand is blue, shoreline is orange. So you can see that shoreline dissolved oxygen tends to be lower. If I showed you the error bars on this, they would overlap, but this is just sort of a first cut. We do see differences in oxygen as we go from open water, from the stand, the most sparse part of the stand to the most dense part of the stand. And this has ramifications for fish. So again, back to this sort of overview of Big Marine, shorelines here, open waters here. You can see us out here in the open water. They did some drone, got some drone footage for us last summer. So dissolved oxygen is increasing from shoreline to open water. And we do see differences in the fish community. So we're seeing this a little bit here because we have 10 minnow traps, and we keep track of what kind of fish we catch where and the numbers of fish we catch in which trap. What we see is that there tends to be more of these sunfishes and mud minnow, which remember are tolerant to low oxygen, in these more interior areas. And if we see minnows, so species that are in the family of minnows, they are only present at the very edges of these sparsely vegetated edges of cattail regions. And remember something I told you in the beginning, which is that for the fish that humans care about catching, this is what they primarily eat. They eat more minnows than these species, partly because they're easier, they're not so spiky. They also prefer more open water zones with sandy substrate because minnows are a species that school to avoid predation. So that's their main anti-predator strategy is schooling. Whereas if you're a sunfish, you tend to hide in vegetation. It has to do with the way they swim and their body form. So I'm very interested to see if we'll see more minnows in the cattail remove site compared to our cattail retain site. So that's just something in terms of fish community I'm really interested in looking at the within, the between cattail retained, cattail removed data. Just a summary of what impacts we expect or might expect. So of course, our goals are to understand how cattail removal affects fish communities and how this varies at least somewhat regionally in Minnesota. We want to determine if cattail removal is an effective management strategy, and more importantly, this sort of smaller scale cattail removal that could be done at the scale of a landowner. So the idea here is to directly inform the DNR to help make landowner permitting decisions. So if these openings could be beneficial, there might be a way to create some more of these openings at the landowner scale, which would perhaps increase, have some ecosystem value spread across a whole lake. We also really have an important and robust public outreach component. We don't want to just say, hey, remove your cattail. We want to say the point of removing your cattail is to reestablish and restore these native plant communities that have that physical variation that's more beneficial to fishes and bugs and amphibians and probably also birds and other species. So it's important to us that we get that idea out, that this is sort of a restoration, not just a removal of cattail. And I think you could probably find some creative ways to permit that as well. This is just an example of a postcard-sized informational thing that Kristen Lobeek, who is no longer at MASERC, but she was their outreach coordinator their publicity coordinator, and she helped us develop this postcard. So we'll be printing some of these and handing them out on the boat launch. When you see people, and you encounter the public in the field. Lastly, some future research. Where do we want to go next? Because even though we're not done, there's still a ton of questions. I think it'll be really important to monitor cattail regrowth across the years. How long do these openings persist? And how much maintenance would they require to keep them open? Would a landowner be able to just zip around with a little underwater weed whacker, every year and clean up the edges and keep these areas open? So what maintenance is required? Maybe rectangles aren't best. There's some work in the Great Lakes looking at circles and other configurations, maybe some connections, U-shaped connections through the marsh. What might be useful. As I said before, mowing cattail on ice, we did that this year at one site because we needed to. It'll be interesting to see what that looks like in the spring, to see if we'll have to go back and clean that up or not. I'm just not sure. Floating mats is a big issue that we did not deal with. This also gets into how old these stands are. Floating mats, as you probably know, are a huge problem in Minnesota. We kind of didn't deal with floating mats in this iteration of the project, but I think it will be important to deal with next. Voyagers has been doing some work on that, and I think that's something that probably needs some work on. And lastly, and one of the cool things we figured out in the Great Lakes region is that when you open up cattail, muskrats are super happy. They don't care that it's hybrid cattail. They use it. So in many of our channels, this is Hannah standing at the end of one of our channels, we would find a muskrat hut. So we may try to throw some GoPros out this summer and just see if we see any muskrat activity. It would be really interesting to do sort of a companion wildlife study and see if there's anything interesting that if muskrats maintain openings. With that, I'm happy to answer questions. You can see our funders, Minnesota Aquatic Invasive Species Center and Environment and National Trust Fund. You can find more information at our Sea Grant website or at the MASERC website. And yeah, happy to take questions.
All right, thank you so much. So for everyone, you can put your questions in the chat. I'll read them off. I don't see any right now, but I have some questions, so that's okay.
Okay. Yeah.
What do you see an issue with some of the removal techniques, like with the big cutting boats or anything, causing so much disturbance that there's going to be a hard restoration for the native plants?
That's something we hope to get at this year. So, we wrestled with, do we need to have the exact same removal technique at each site? And what we decided on was it needs to be realistic to what landowners could possibly do. So it turned out that we ended up with the same contractor doing all the removals, but it's really variable what they use depending on site. So they did use some of those big, that big sort of chewing, cutting stuff at some sites, and at some sites they had to actually hand remove. So I think it will be interesting to look at. We didn't do any removal of sediment. So this was all just cutting below the surface. And so I think that's something we'll have to think about when we go back and look at regrowth this year.
That'd be interesting. Could you talk about how the Minnesota DNR and Bowser view the removal of cattail in the design you discussed, and how they have responded to permitting requests?
Okay, yes, I can talk about that as much as I know. So I would say for us, I guess, do you mean I guess I'll just talk about what I know. So what I know was, right now you can request, for us, we have a research permit. We did need to do a lot of outreach work with DNR to talk about our study and talk about what we were doing. We originally thought about doing this on landowner property with buy-in from landowners, but DNR was more comfortable with us doing this on public land. So all of our sites, with the exception of one, is on public land. So they have been very supportive for our research. We have a research permit to do this, and that as well as an aquatic plant management permit, and they have been great and very supportive. I think they are waiting to see what we find, and I don't know how they will respond in terms of permitting in the future. It depends on what we find in our study. One of the things that's important to consider is in our openings, we don't have any human use. I mean, we have a lot of fish sampling, but we don't have boats going in and out. So I think that's going to be really different in terms of, are you going to allow permits just for maybe a restoration swath, and also a swath where you can use your boat, because if you're disturbing it all the time, maybe you won't get that native plant regrowth. So I'm not sure I answered that question. For us, it's been, they've responded really well to the study. I think there's some concern that if you start removing cattail and telling people it's a good idea, they're going to be inundated with requests for that, and that people are going to not respect it, and it's going to cause a big problem. The flip side of that coin is it already is causing a problem, and people are going to their legislators, and that could be regulated that way. So I think we're kind of in a sweet spot of, like, it's important to get this information and get a handle on it. And so the DNR has been supportive of our study. If it didn't answer you, you can put it again in the chat.
Yeah. Okay, so along the same lines there, how do you think those agencies, or even the U.S. Army Corps, would respond to using more hydrology management, whether you change the wetland type to a wetter system or install a control structure for this cattail management?
I'm not sure. I guess I would, with not having dealt a lot with Army Corps of Engineers and that type of stuff, I would guess that that's probably not the way to go. I mean, what we see, a lot of people use that water management to manage things like waterfowl. We actually run across it in Voyagers, because they manage those lakes, right, with water level, and that it actually makes it really complicated. So I can see that in the sort of prairie pothole wetlands, where you have maybe can restore that flood dry season. I think that would help handle cattail. But if you increase water level and make it sort of more stable than it is, it probably isn't going to help. What you need to do, and for lakes, that's not really going to be an issue, right? Because it's a lake, so there's water. But in those more ephemeral or those wetlands with that really clear, wet, dry, regulating that is bad for. Let's see, it's bad if you don't want hybrid cattail, right? If you keep that change of high, low, you're going to be less likely to have hybrid cattail. But if you make that water more permanent, that's a problem. Does that make sense?
Yeah, I always tell people who complain about cattails around me that humans and cattail have a lot in common. We both like steady water levels.
That's exactly right. That's totally exactly right. Yeah.
Yeah.
If we could change it, it would be better.
a real quick insight. Someone from the DNR, Jenny, says she could give a little bit of the DNR perspective. Jenny, I allowed you to unmute yourself if you wanted to do that.
Awesome.
I think the first question was how DNR, and I can't necessarily speak for Bowser, though I think Bowser has less regulation over the removal of cattails likely on those WACA wetlands. But you know, within Minnesota DNR, we have our public waters rules that are quite strict, and those are going to only come into play if there's going to be disturbance of the sediment, or, you know, you're going to change the bed and bank. If you're going to do removal of cattails in some other kind of manner, typically that would happen through our aquatic plant management permitting, like you said, and we equally have a strong interest in learning about that. And so beyond a certain area, then we have to look at doing an individual plant permit. And you're right, it's easier right now to do a research permit because we would have to change our rules if we wanted to make these sorts of things easier, and that is opening it up to the legislature or sending it out for public comment, etc. And so I think that we are constrained by our own rules and statute, right? And so we are working inside of those. But many people at the DNR from across divisions, Fish and Wildlife and Ecological and Water Resources, we all do have a strong interest in understanding this better. And of course, we manage so many lands and have a strong interest in managing our own lands the best that we can, and that includes cattail management. And then just as far as then that next question, you start to get into that public waters work permit and a whole different set of rules where you're really talking about managing water levels. So things like water control structures, then that has to get a different kind of public waters work permit, and you've got to have a responsible entity to manage that and to really know that whatever management plan you've got in place is actually going to achieve the goal you're going for, right? And when you're installing some kind of a control structure, almost always there are the adjacent landowner concerns. When we've got these big land areas like Voyagers or some of our larger state parks or some of the Fish and Wildlife Service areas, we are able to manage those wetlands without having to worry about neighbors so much. But there's many basins where you just don't have the freedom really to manage the water level in the way that you'd like to get at your goal of managing things like cattails, because there's so many competing interests. So that's kind of our view on it. And our rules are very restrictive for putting in new control structures and for the uses that can be approved for control structures. And so I think similarly there, we would end up having to go and change our rules or statutes again. Not to say that that wouldn't ever happen, but for sure we've got some constraints in place as things are now.
That's really good insights. Thank you.
Yeah.
Okay, well, we have one more question here. I have plenty because I'm trying to work on a cattail project right now, but we don't have to go forever. But do you have any recommendations for cattail control on floating mats that also have native veg? And how do you prevent degradation and infringement over time?
Okay, so I do not have a lot of direct experience with floating mats. So can you just read that again? Do I have floating mats and native vegetation within a floating mat?
Yeah, so I reckon maybe
Let's see here.
a floating mat that might naturally exist is how I read it. And and maybe cattail is growing into that, maybe bog mats that are kind of becoming cattailed or something like that.
Yeah, I don't know. I don't have a lot of experience with that. What I can say about floating mats, you know, and really my experience with floating mats would be just strictly cattail floating mats, right? Those older stands tend to really extend into floating mats. Voyagers has been doing some work chopping them up, and what they're doing is and they spend a lot of time doing this. It's a lot of work. They chop them up, they put them on shore because they don't have to worry about landowners. They put them on shore, and then they actually reseed that biomass, cattail biomass, with native vegetation. So they're trying to actually, when you have a floating mat, you almost always have also a lot of cattail in the sort of wet meadow zone, right? That's not really the lake. So they take all that biomass, they smush down the cattail that's on shore with that biomass, and then they're reseeding it with native vegetation to try and get that vegetation to regrow. I don't know. I think it would be very complicated to try and remove cattail from a bog mat that's a whole bunch of other vegetation, short of digging it out individually. If you have some hybrid cattail and you can get rid of it before it expands, that's better. I mean, what we learn is that the earlier you can, the shorter amount of time a stand has been there, the easier it is to remove those longer stands. The stands that grow and grow and have been there for a long time present a real challenge. So I'm not sure I really answered that question, but.
I think it's good. Maybe one more. So a lot of your research has been looking at the cattail that are pretty much in that zone of depth where they're maxing out. And so you can kind of cut them under the water and everything. But as you mentioned, you can see them up in that wet meadow area, and a lot of these urban areas, they're pretty dominant in kind of that real shallow emergent, or they've terrestrialized enough so much that it's almost above water. And so you can't even cut them below the water line.
Right.
Do you think that there's a future for or how do you see maybe repeating management that would suppress and stress out the cattail that would be feasible and increase that species diversity?
Yeah, so I think a couple things, you could look at mowing. I mean, and all of these, it's so much work, right? You have because what you end up with is a whole bunch of biomass you have to deal with. So in an area where you could use some of that sort of biomass you take out of the water to crush or smash down the live cattail and try to regrow like Voyageurs National Park does. Mowing repeatedly does seem to help. There's some work in the Great Lakes region in northern Lake Michigan where they did some studies of sort of crushing versus mowing, and those things can suppress it. You have to kind of repeat that every year, right? Because it does grow back. You just have to kind of keep taking that biomass out. And they have shown that after years it does help. They've also shown that sometimes you end up with a lot more methane release after you do some of these techniques. So in terms of greenhouse gases, it can be problematic. But I would say mowing, crushing, and actually trying to reseed native vegetation on top of that dead biomass. So using that dead biomass as sort of fertilizer, right? Because one of the issues when you have all this biomass is where do you put it? Because as soon as it decays, you're going to get a whole bunch of nutrients back into the water. But if that could be used for reseeding and regrowing native species, that's something that I think Voyageurs has had quite a bit of success with. So that's an option.
Yeah, that's interesting. Jenny adds in the chat that Sheil Bansal has done some neat research looking at cattail management and climate change. So check that out.
Yeah, there's a review in 2019. There's a big review paper in wetlands on Taifa that Sheil led.
Anyways, it looks like to be all the questions we have. Really appreciate you coming in and presenting. This is a neat topic, and I think there's a lot to learn still. So good job security for you, I guess.
Yeah, there you go. Can maybe keep getting funding.
Yeah.
Thanks very much for the invitation. I appreciate it. And thanks everyone for your questions. I really enjoy it. I enjoy all the questions because there's a lot. I'm a fish biologist, so there's a lot I still need to learn.
Great. Well, we
Cool.
appreciate it. And till next time.
Okay. Thanks everyone.
All right. Bye all.